My name is Simon Travers. I'm CEO of Hyrax Biosciences, and I'm joined this evening or today by my colleagues Imogen Wright and Natasha Wood. And we're excited to share with you our solution which deploys SARS-CoV-2 lineage clade typing through Exatype. And as you'll see, Exatype is an easy to use online DNA analysis platform. So very briefly in one slide, I just want to give you an overview of where we fit in the genetic diagnostics. We specifically operate as a company in the sequencing-based genetic diagnostics space. And we see sequencing-based genetic diagnostics as essentially a three-step process. The first step being sample prep, so taking your sample and preparing it for sequencing. The second step being the DNA sequencing step itself, whether using next generation sequencing approaches or Sanger-based approaches. And the third step being the data analysis and interpretation. And depending upon what type of diagnostic you're running, the reporting for that interpretation as well. Whether that be, for example, as we're talking about tonight, genotyping and specifically lineage and clade typing, or right through to drug resistance or drug susceptibility testing for oncology or for infectious diseases. So Hyrax very much plays in step 3. We have developed Exatype, which is a software solution, a disease agnostic DNA analysis platform. And essentially, Exatype takes raw next generation sequencing or Sanger data as input and automatically undertakes both the analysis, interpretation, and reporting, regardless of what test is being performed. And so, as you'll see later, Exatype can be deployed across a wide range of sequencing-based genetic diagnostics of scale. And for us, the ultimate goal for diagnostic software is that actually at scale there should be no need for end user interaction. So, you know, for those of you that are experienced with software analysis, you'll see that a lot of the software solutions that are available are either incredibly complex or complicated to run, or in fact have a lot of manual steps in running the data and running the analysis. And really for us, the ultimate goal is that at scale there would be no need for end user interaction. And particularly because manual interaction unnecessarily introduces the threat of human error, but also impedes scaling. If you have somebody needing to interact with the software for every reaction or every analysis, then you're impeding the ability to scale. So if we're able to use robots for sample prep and we're able to automate DNA sequencing, then why not automate the analysis and interpretation of DNA sequencing-based diagnostics? And so we as a company have developed Exatype with this in mind. So we're already doing this with a number of customers. We have solutions out there in the marketplace whereby the customers are automatically integrating their sequencing machines directly with Exatype, pushing the data through, the analysis happens, and the results are presented for them to populate the limbs. So essentially, it's a completely hands-off solution whereby end users don't need to interact with it. And so that's kind of the ultimate goal of the software, but we have various grades of the software coming back in terms of the amount of interaction that you want or need to have with it. So what we're going to show you today is a solution of how we've deployed SARS-CoV-2 lineage or clade typing through the Exatype platform. But really, as you see, what we strive for with our software is really to make it as simple and intuitive and as easy to use for the end user. And so firstly, Natasha, my colleague, our CSO, is going to just maybe give a little bit of an overview about the minefield that is currently SARS-CoV-2 lineage and clade typing. And then Imogen is going to provide you with a demo of the software solution.
Thanks, Simon. So as Simon said, my name is Natasha, and I'm just going to take a couple of minutes to highlight some of the current standards for sharing SARS-CoV-2 sequence data, and as well as the tools for defining clades, lineages, and variants, which will provide some context before Imogen then goes through a demo of the Exatype platform. So from the beginning of the pandemic, sequencing efforts have been massive, and these continue to scale. And this has allowed global surveillance of how the virus had spread and changed over time. And as of this morning, over 460,000 genomic sequences have been submitted to the global repository GISAID. So you can imagine with this amount of sequence data that we need to find ways to group and define viruses in order to communicate and track transmission. So all of this data, all of these 460,000 sequences can be represented on a phylogenetic tree or a schema such as this one. And this image is taken from Alm et al. In a publication from last year. And what it represents is every sequence available in the public repository grouped in different ways. So for SARS-CoV-2, there are two commonly used terms or methods to group the sequences. And these are clades and lineages. And for each of these, Nextstrain is used to define the clades and Pangolin for the lineages. And these tools, really what they do is that they zoom into the different groupings of the viruses at a different scale or resolution. So just briefly, you can think of clades as a broad classification where sequences are grouped by combinations of signature mutations. And I'm sure you guys will be familiar with this Nextstrain graph. So over here, you can see that so far 12 clades have been defined, with the first clade 19A, which denotes 2019 and A for the first clade, as well as 19B. And then from 2020, you get from A all the way down to 20J501YV3. So this represents the clades for the entire SARS-CoV-2 pandemic. The next resolution or term is lineages, and this is a finer scale. And lineages do something slightly different. They can capture any of the sudden dominance of a specific lineage. So if there's suddenly a lineage that grows faster than some of the other lineages or sequences, then you can see this at the front edges of the tree. And lineage definitions are particularly useful for tracking location-based outbreaks. And again, if we look at a global tree for SARS-CoV-2, which is created by the tool called Pangolin. Over here, you can see where it starts with a broad hierarchical clustering. As these edges of the tree evolve, further subgroups are defined. So that's just some quick background on how Nextstrain, clades, and Pangolin lineages are used to place any new genomic sequence within the background of almost 500,000 sequences currently in the global repository. And I would like to point you to a brilliant presentation if you want to dig deeper into the definitions of clades and lineages, which was part of the Arctic Network and Climb Big Data workshop in the middle of January. The final point that I want to mention are variants. So individual mutations or constellation of mutations can impact biological phenotypes, such as transmissibility or infectivity. So for example, as shown here, and which was one of the first variants that made global news, is D614G, which is in the spike protein. So where D was prominent at the start of the infection of the pandemic, G at position 614 quickly took over and became prevalent as the virus spread. So together with the groupings of clades and lineages, these variants provide different kinds of information and can be linked, as I just said, to transmissibility and infectivity. So the tool for defining variants is the same as for clades, and this is called Nextclade. And Imogen is going to go through this a little more detail when she does the demo, but along with your sequence name, here you have your QC, so quality control, the clade definition, as well as both the nucleotide and amino acid substitutions or variants. So just to bring this all back together into the context of the current global news and surveillance, we're all familiar with the variants of concern that were first reported in the United Kingdom, South Africa, and Japan from persons from Brazil. And each of these were first locally referred to as respectively VOC 2020, so variants of concern 2020-1201, and the South African one 501YV2, and the one that is now dominant in Brazil as B11281. And each of these have associated Nextstrain clades pangolin lineage and defining variants. So for the sequences that first identified in the United Kingdom, which is now commonly referred to as B117, it has the associated Nextstrain clade 20I 501Y B1. And of the 17 defining mutations, I've just highlighted 501Y over here, which confers a stronger binding affinity to ACE2. And then D614G, which I mentioned before. So for both the variants first identified in South Africa, 501YV2, as well as P1, which is now dominant Brazil, which have their associated Nextstrain clades, each of these over and above 501Y also have E484K, which has been associated with reduced susceptibility to neutralizing antibodies, which has been worrying in the news in relation to vaccine efficacy. So if this all seems quite confusing, don't worry, it is. And while there is a drive to define a standard nomenclature for describing these variants of concern, for now, both the Nextstrain clade and the Pangolin lineages are commonly used. So with that background, I'm very excited to hand you guys over to Imogen, who will take you through a practical demo of the platform to show you how we've incorporated these tools into Exatype.
Okay, I hope you can all see my screen. So this is what Exatype looks like when you land on our dashboard. And the first thing I want to draw your attention to is sample runs available over here. So what this says here is that I have 44 SARS-CoV-2 sample runs available, 44 credits essentially, and each of those corresponds to one SARS-CoV-2 DNA sample that I can analyze. You'll notice that next to the SARS-CoV-2, there's a little plus button, request another pathogen. Exatype is a platform, and while for this presentation, we're going to be focusing quite heavily on SARS-CoV-2, Simon has hinted, and we'll discuss it a little bit more, that there are a lot of other things we can do. So there are two main sort of tasks that you have in Exatype. You can either view existing jobs or you can create a new job. A job is essentially an upload of DNA sequencing data from a DNA sequencing platform that you want to analyze to produce a result. So I'm going to take you through creating a job now. So I'm first going to name my job. I'm going to creatively name it webinar. And then you have some optional details that you can add for your reference to this job. So you might want to say that actually this data was only sequenced on the 28th of January. You might want to say it was sequenced using the Arctic protocol, but this is completely free form. You can say anything you like that'll help you understand what this job is about. Next, we currently support 2 sequencing platforms with a couple more on the way. We support IronTorrent and Illumina data. I'm going to upload some Illumina data, and I'm going to upload paired end, not single read data. This next screen is a little bit interesting. So this is where you get to define what primer scheme you used to sequence your data. You do that by means of selecting a bed file. A bed file is just a way of describing a primer scheme. It describes how you essentially extracted the DNA, amplified it, sequenced it. The important thing about this part of the process is that in many other pipelines, it's a manual step you have to do before. And it's very important because only data in each of your DNA sequencing reads that lies within the inserts of your primer is real DNA data. The data outside that is actually part of the primer. And if you don't correctly trim off your primers, what you can get is essentially fake wild types. You can get primer data mixed in with your real data that then dilutes your wild type down to, dilutes your variants down to a level that it's not detectable. This is often a manual step in other pipelines, but we just automate it away from you. In our public platform, we have 3 versions of the Arctic protocol. I'm going to upload some Arctic v3 data. We have partnerships with other assay manufacturers that Simon will touch on in a moment that will allow you to use other primer schemes from other assays as well. So now we get to the crux of the issue, choosing your sequence data files. So here's some data. It's really simple. You just select it. You can even select some junk at the bottom. I've selected an Excel spreadsheet. And all it does is auto-detect your sample name. If you don't like the sample names that it's auto-detected for you, you are entirely welcome to fiddle around with them as much as you like. If you don't like how you fiddled around, you could reset them, but I happen to like that one. And also it'll tell you that I've uploaded demo data details.xlsx, which is not a valid FASTQ file, and it'll just completely ignore that. So we try and make this really as intuitive as possible, sort of, you can't stuff it up. And that is just about it. So I'm now going to click here to upload files and run your analysis. And off it goes, uploading. What's great is you can now more or less walk away, go have a cup of coffee, and that is the end of your hands-on involvement in this process. What it's going to do in the background is it's going to do all your quality trimming and quality analysis for you. It's going to do all your primer trimming. It's going to map to a reference, call variants, generate a consensus, and do your lineage clade typing for you. So there's that job running. I'm now going to show you one I ran earlier just to save a little bit of processing time. And that's over here. So this is a 14 sample job that I ran earlier. So these are the kinds of results that you get at the end of the process of running Exatype. So that was your whole upload process. Now let's see what you get back. The first thing I'm going to take you through is a little bit of quality control of what your sample run looks like, and just some things that you might want to kind of look at to make sure you're doing a pretty good job of doing your sequencing. So the first thing we give you is what we call a fateable reads plot, which is essentially per sample, how many reads map to the SARS-CoV-2 reference paired, or just the forward or reverse side mapped. So this yellow is map paired, which is kind of, you want to see as much of that as possible. Then you get data that is very low quality, like here in sample 1, there's quite a bit of it. Low quality data means that the whole read was marked low enough quality that we considered it to be junk and threw it away. Then you get not aligned reads, which often represent contamination or just, again, bad quality data with a lot of mutations that doesn't map. So this is kind of a good way to say this is a pretty good sample run. The data is reasonably evenly distributed across the samples, and most of the data in the case of most samples did in fact map to the reference. So that's one first tick that we've done a reasonably good job of sequencing here. Then the next tick we want is the read coverage of the SARS-CoV-2 genome for all samples. So along the x-axis here is just my position in the SARS-CoV-2 genome. And here you get for the entire run, your minimum, maximum, and average coverage. So your maximum coverage is floating around sort of 14,000 at the top. And minimum coverage, there are a few dropouts that you can see here, but your mean coverage is actually pretty good. You're well into the thousands. So this is actually a pretty good sequencing run that we've got here. You can actually see the Arctic primer scheme doing its magic here. There's a little bit more coverage in the middle of the primer insert and a little bit less on the edges. That is expected. And then you can see some primers are a little bit oversubscribed and some primers are a little bit undersubscribed. That is also expected. So we can kind of get to the end of this part and be like, okay, we have a good sequencing run. All our samples are reasonably well represented, and we can start digging into some actual results. So I'm going to go down here. These are individual results per sample. I'll get into some aggregated results in a minute, but this is kind of the crux of what you want to see for your samples. So the first thing we give you is a last little bit of QC, which is your sample level coverage plot. So this is how each primer was represented across the genome for this individual sample. Next, this is the part where it gets, for the purposes of this webinar, really fun. We give you your lineage and clade typing. So as Natasha very well explained, you get a Pangolin lineage, which in this case is D2, and a Nextstrain clade, which in this case is 20F. What you will also get out of Nextstrain is a little bit more QC information. So over here, you can hover over this little tick, which says this is a clade that we're pretty confident about. And it's going to tell you there wasn't much missing data. There weren't very many sites that were mixed. So where it's sort of an AG mix or something along those lines. Private mutations, so mutations that you wouldn't expect in SARS-CoV-2, or SNP clusters, where you've just got a large number of mutations in a small area, which might indicate bad sequencing. So not only do you get, okay, this is my clade and this is my lineage, but you get a kind of additional QC confidence measure there. The next thing that you get, as Natasha was saying, is your amino acid substitutions and deletions. So this comes straight out of Nextstrain again. And you can see here in this case, in the spike protein, I have the classic D614G. So this is not a variant of concern, but it does have a D614G, which is now the dominant mutation worldwide. You will also get here a consensus sequence that you can download. You can upload it to a database, you can do downstream processing. And just in case you're one of those people who likes to visualize your alignments, we will also give you the consensus aligned to the reference. And finally, we'll give you all your nucleotide variants and their positions. Then I'm going to show you, just for fun, a more interesting one down here. So this is a 20H Nextstrain clade, a B1351. This is 501Y-V2. I'm currently in an Airbnb in Cape Town for a lot longer than I thought I would be because of 501Y-V2, so it's personally relevant. And here you can see it has both N501Y, which increases ACE2 receptor binding affinity, and E484K, which may reduce the efficacy of broadly neutralizing antibodies. So this is exactly the kind of profile that you'd expect for a 20H 501Y-V2 variant. And here you've also got the classic spike protein deletion. So that's kind of what you get on a per sample basis. I'm now going to show you, if you just want to get a quick aggregate. Here is all your lineage clade typing together. You can scroll down, just have a look at it all. You can see here a few where there's a little bit of missing data. So Nextstrain is a little bit less sure about these. Here's one where it's a lot less sure about these. But in general, this is a pretty good run. And you can see there's quite a lot of variety in the clades and lineages being produced. This is a really nice overview for the humans. A nice overview for the computers is present at the bottom. So this is downloads. So here we can get all consensus sequences from all your SARS-CoV-2 samples, which you can then upload directly to a public database such as GISAID. They'll do any downstream processing that you like on it. We'll also give you all the variants in all of your samples that are present at more than 10% prevalence, so you can do analysis on that. And finally, we'll give you all your raw Nextclade and Pangolin files, which have essentially the computer readable version of this information that you can then again do any downstream processing that you like. It will even give you your Nextclade tree. So that's essentially an overview of what you can get out of a SARS-CoV-2 lineage and clade typing job. I'm now going to hand back to Simon, who will give you a little bit more detail about Exatype as a whole and the platform and what you can do with it.
Thanks, Imogen. That was awesome. So just to close it out, I very quickly just want to kind of just make a couple more points. So one of the key things to see is that Imogen has been doing that upload and has been processing the data through a website, and we are currently hosted in the cloud. And what that means is we have deployed Exatype as auto-scaling architecture. And what that means practically for you is that when you upload data to your account, or you do a run, as we call it, that is given its own dedicated computer. And what that means is that essentially we have unlimited scaling in the cloud. So you could have hundreds of labs, thousands of labs across the world running data at the same time, and you will see zero impact on the speed or the time it takes for your results to be presented. So Exatype is fully deployed as a kind of a modern scaling solution. And as you've seen there, and I hinted at it earlier about is kind of enabling high throughput analysis, and really what we've done with this web-based solution is we've deployed a solution that enables high throughput analysis of SARS-CoV-2 data. So rather than needing to take your FASTQ data, manually process it, upload it to Nextclade, upload it to GISAID, Pangolin, we essentially have taken that entire process, as you've seen, and enabled it just to be essentially a single click process that I hope you agree is easy to use. And that is how we've deployed the SARS-CoV-2 solution, but Exatype does have various other options for use. And so we actually have built APIs on the front end, which enables direct integration with sequencing machines. And I hinted at this earlier. We actually work with customers who once their sequencing machine produces the raw data, that essentially can get pushed directly through to Exatype with no need for manual intervention. And that it just essentially means that the data can be run automatically. And we do also offer local deployment. And when I say local deployment, there's essentially two ways of being able to do that. You can either run, we can ship you the software, and you can either run it locally, using the graphical user interface, so it runs on your browser, or alternatively, there is a high throughput solution where you can actually run it on a high performance cluster. So there's various options around how you can use Exatype, and primarily that's to enable high throughput analysis that is relevant for you and relevant for your users. And then in terms of outputs that we produce, so Imogen has very well shown you through the kind of the manual results that you can look at, the clear actionable reports that you can browse and look at on our web interface. However, as she's also shown, we do make outputs available in standard formats that you can download for kind of further analysis. So for example, the consensus sequences that we produce per sample can be used to be used to directly upload to GISAID. They can also be used to drag and drop onto the Nextstrain or the Pangolin web-based services, so that you can do whatever further analysis you'd like to do there. So really what we try and do is we try and have the outputs available in as many formats for kind of continued use. And then one of the kind of the other key things that we do is actually we have an API in the back end as well, which enables direct integration with LIMS systems. So essentially there is a way where data can flow directly off the machine into Exatype. The analysis can happen and the results can be presented to an end user LIMS with zero end user interaction. And that's really where we're striving to go with Exatype as a product, but it is possible to be done on many solutions. And one thing to point out is that Exatype is essentially an extensible solution. So we have designed it in a modular fashion, which means that it can be customed in a broad range of ways. So when Imogen did the demo on a kind of our public front-facing solution, you saw that we had support for the 3 Arctic protocols. However, in the background, we do support a number of other protocols, and I'll touch upon that in a moment. But technically, it can be deployed to support absolutely any assay. So for us to introduce support for an assay is actually quite trivial. And then also the default output. So again, what you saw in the demonstration was our default outputs and our default formatting. However, if you have customized information that you'd like to present, we store all of the results. We store a huge wealth of information about every sample in the background, and it is very easy for us to then deploy different outputs, different formatting on the outputs, etc., either on a per account basis, or theoretically in a site-wide manner. So if you've seen something this evening that you like the look of what we're doing, but actually you think that from your perspective, and you think from our users' perspective, that it would be good to present our results in a different way or present different types of results, we can just make these changes trivially and push that through in a site-wide manner. So we're very flexible in terms of how Exatype can be customized. And I think the key one here is that it can actually be offered as customized companion software. And so Imogen did mention that we've some partnerships in a number of solutions, but in the SARS-CoV-2 solution space, for example, we've partnered with Paragon Genomics. They have produced a step 1. They have an assay for sequencing SARS-CoV-2, and essentially we are able to customize Exatype to support their product. And what that means is we support their assay, but actually we can also do customized URLs, a dedicated solution for a particular assay, a customized look and feel, branding, unique reporting, etc., and outputs. And so actually, while you've seen our public kind of front-facing solution, there are other alternatives and there are other options in terms of how we can deploy our software as companion software. And just to step back just for 2 slides, just in terms of, just kind of give you an oversight of Exatype as a platform. So we've very specifically spoken about it as a SARS-CoV-2, clade and lineage typing solution. But really, Exatype is, as I mentioned earlier, a disease agnostic solution. So we think of the underlying platform as being the algorithms drive that platform to power it as a solution. And essentially, we develop what we call plugs that fit in the top and utilize the power of the platform to convert it into a disease-specific or an application-specific tool. And so these algorithms that I mentioned, we have developed a suite of novel algorithms that power the analysis, interpretation, and reporting within the Exatype platform. And particularly, I think, pertinent for a lot of genetic diagnostics is that actually our algorithms have been customized for very accurate detection of low abundance variants in heterogeneous data. And that is particularly pertinent for some types of diagnostics. For example, we've done some work in cancer that shows that these algorithms reduce the false positive rates in profiling of breast cancer tumors from a median of 16% down to less than 1%. So you can understand that from a clinical perspective, this level of accuracy is incredibly important. We've also shown similar results for HIV drug resistance testing as well. But I think the key thing about the platform is that it can be deployed as companion software across a broad range of sequencing diagnostics. So all the information that harnesses the power of the platform is contained in the plug, and that's what makes it applicable to a particular setting. So for example, Exatype can be very easily deployed for genotyping and drug resistance testing. So you've seen our SARS-CoV-2 solution. For example, we have a couple of HIV drug resistance testing solutions as well, where essentially we can harness the power of the platform to be able to identify variants, call variants, and actually output reports that can be passed directly through, or that are passed directly through to clinicians. We also have the ability to support multi-target diagnostics. So for example, infectious disease panels, distinguishing the presence and absence of various infections. And then, as I've kind of spoken about previously, tumor profiling and drug susceptibility in the oncology space. So you can see there's a broad range of the types of solutions that can be deployed within Exatype, and it's kind of an ever-expanding set of capabilities. So if you have a particular solution outside of SARS-CoV-2 that you'd be interested in deploying, please reach out and we can discuss it further.